Hello, hope you are doing well. I'm going to finish up the second half of the Chapter 15 lecture in this recording. Before we continue, if anyone is watching these videos, I've noticed that some people haven't been submitting their reading outlines for the chapters. Don't forget that those are still due. Also, again, there is a worksheet associated with this chapter. Go ahead and check that out on Canvas. And if there's any problems that you're stuck on, there is a survey where you can submit questions that you want me to go over. Make sure that you do that in time so that I can help you out the best I can before that assignment is due. So let's talk about Objective 3, the secretory pathways used by the cell. We've talked about these a little bit so far. We've talked about how vesicles are used in the secretory pathway. So we know that proteins that are going to be secreted by the cell are made by ribozymes on the surface of the endoplasmic reticulum. So those proteins to be secreted, as they are being synthesized, they are inserting immediately into the endoplasmic reticulum. And they are then going to go bud off of the ER as a vesicle and travel to the Golgi and so forth. Now, in addition to being inserted into the ER also has some roles in modifying proteins. So after they've been synthesized, there are some post-translational modifications, right? Post-after translation, creation of the protein. And so these are usually attached onto proteins by covalent bonds. One example here that we have is glycosylation. So glycos, glucose, sugar, right? So we have these groups of oligosaccharides that can be attached onto the protein via a covalent bond. And there's different patterns of different sugars, molecules, and chains and branches that can be attached to different sugars. And so the main thing from this slide that I want you to remember is that in the endoplasmic reticulum, you can have modification to proteins by covalent attachment of sugars. Check out this video. I encourage you to watch it. It talks about covalent modifications and functions of the endoplasmic reticulum and so forth. So covalent bonds, what type of covalent bonds can we have? So there can be, in addition to attachment of these sugars, you can also have disulfide bonds, so helping with the formation of maybe a more complex tertiary structure of a protein. Again, here, this example we showed was attachment of an oligosaccharide. Now, what are these oligosaccharides and these disulfide bonds doing? So again, we've talked about disulfide bonds. They can be within a chain of a protein to help form a certain shape, or it can help to connect two different chains together to form a larger complex. Oligosaccharides have a variety of functions. First of all, they can help protect the organelle, or if it's going to be sent out to the surface of the cell, it can help to protect the surface of the cell. This web of sugars kind of acts like a buffer. They can also help determine where this protein in this vesicle is going to go. So it is a modification that is kind of like a tag, kind of like a signal peptide, but in this case it's a sugar that's added onto the protein, and it's kind of like a barcode to help the cell know where to send this protein. If this protein gets sent to the surface of the cell, it can be used for cell surface recognition. So cell-cell recognition, if a cell sees another cell, it says, are you familiar? Are you part of me, this body, right? So when we talk about immune rejections of transplants, it has to do with these patterns of sugars on the surface of the cell. So different people have different patterns of sugars, and it helps the cells to recognize self versus non-self. Now, attachment of this oligosaccharide, again, we said only happens in the endoplasmic reticulum, and it is completed by these enzymes called oligosaccharyl transferases. There can be further post-translational modification in the Golgi apparatus as well. So protein has been made, it's gotten some covalent modifications on it to kind of tweak it, and it's getting ready to leave the endoplasmic reticulum. Now, before that protein can leave, there is a checkpoint in which the cell can make sure that protein has been formed correctly. Right? Misfolded proteins are no good. You've probably heard of different types of diseases where proteins are misfolded. They can also, when a protein is misfolded, and in addition to not being able to function properly, it's also going to aggregate. So it's going to find other misfolded proteins and form these big clumps, and these clumps can actually impede normal functions in the cell and cause toxicity. So there are chaperone proteins in the endoplasmic reticulum that will help to make sure that these proteins get folded correctly. So if there is a misfolded protein, the chaperone protein grabs onto it, right? Just like at a high school dance, if there's someone trying to spike the punch, the chaperones are the ones that are supposed to catch that miscreant, right? So the chaperone protein is going to hold that misfolded protein and keep it from leaving the endoplasmic reticulum. So if the protein is fine, if it's folded properly, it's not going to be grabbed by the chaperone, and it can bud off in a vesicle and travel onto the Golgi. Again, proteins that are misshaped will aggregate, they will bind to each other and clump up. And there are also proteins that can, if it's misfolded, if it gets in contact with another protein, it can cause that protein to be misfolded as well. So it can kind of spread. So some of these diseases that have to do with misfolded proteins and aggregates would be things like Huntington's disease and prion diseases. And these chaperone proteins not only keep the misfolded protein in the endoplasmic reticulum, but they also direct refolding to help fix this protein. How that works is a system called the unfolded protein response, or UPR. Right? We have these sensors that are on the surface of the endoplasmic reticulum, and they're facing into the ER, and they are sensing for misfolded proteins. If these sensors, if these proteins identify a misfolded protein, they are going to induce a cascade of a signaling cascade that ultimately involves in the upregulation of transcription factors. And what do transcription factors do? If you remember, transcription factors regulate whether a particular gene gets turned on or off. So there are chaperone genes and other types of genes that make proteins that help to refold proteins that are misfolded. And so these sensors are going to send a signal to increase the amounts of transcription factors. These transcription factors that are increased are specifically targeting chaperone proteins and other proteins in this repair mechanism, which is pretty cool. Another thing you can see in this pathway is that one of the sensors actually, in addition to upregulating these chaperone genes, it will also slow down protein synthesis, which makes sense. If the proteins that are being made are not correct in their shape, it's a good idea to say, hey, slow down, stop the presses, we've got to fix something that's not right. Right, so again, this UPR, unfolded protein response, when it senses misfolded proteins in the lumen of the ER, is going to cause upregulation of transcription factors for chaperones that can help with accurate folding. Now, after proteins leave the endoplasmic reticulum, if they've passed all those checks, the next place that they go is the Golgi apparatus. Now, the Golgi apparatus, some people have said looks kind of like a stack of pitas, which I would agree. And every time I show this slide, it makes me hungry for Greek food. But the important things that I want you to note, first of all, is that a protein that leaves the endoplasmic reticulum, if a protein is going to be secreted, starts in the ER, then it goes to the Golgi. That's the next place. The next thing to note about the Golgi is that it has two sides. So the proteins that leave the endoplasmic reticulum always enter into the Golgi on the side called the cis side, the cis Golgi network. And then they always exit out the side called the trans side of the Golgi network. And the way that you can tell which side is which is that the cis face of the Golgi is always facing more towards the interior of the cell, closer to the ER where those vesicles are coming from. And the trans face, or the trans side of the Golgi apparatus, is always facing more to the exterior of the cell because these vesicles, once they get through the Golgi, are going where? Out of the cell. Right? So in this electron micrograph, I've labeled for you the cis and the trans side. Now, let's talk more in detail about exocytosis, right? Secretion of proteins from the cell. So these proteins, once they get to the Golgi apparatus, they can be secreted by two mechanisms that you should know. One is called constitutive secretion and the other is regulated secretion. So in constitutive means like constant or on always. And so that means that as soon as a vesicle is made, it's going to travel to the surface of the cell and be released. Whereas regulated secretion is when the vesicles are made, they've got their content, they're going to travel to the surface of the cell, and then they're going to just hang out and wait. And they're going to wait for a signal. That's why it's called regulated. So only upon receiving the correct signal will the contents of that vesicle be released outside of the cell. So it's really important to understand, to be able to compare and contrast these two types of exocytosis. I would highly encourage you to watch these videos that describe how it works and also some examples that you can use. You should know how to describe an example of both of these situations. So, for example, we've talked about release of neurotransmitters before at the synapse, right? These neurotransmitters are sitting in vesicles and they are waiting to be released, right? And so would you consider that to be constitutive or regulated secretion? It would be the second, regulated secretion. Let's look at another example of regulated secretion. Here we have pancreatic beta cells, and we're looking very up close on this, at the edge of a pancreatic beta cell. So you can see the plasma membrane, the extracellular space is in white, the interior of the cell is more gray, and we have these big black blobs, and that is insulin. So insulin that's been packed really tightly together. And on the left, you can see the insulin is in a vesicle and it's just right at the surface of the cell, but it hasn't been let out yet. And then on the right side, you see a clump of insulin that's just about to leave. The vesicle has fused with the plasma membrane, and that insulin is about to leave the cell. All right. So in this case, this is regulated secretion. So there has to be a certain signal to tell this pancreatic beta cell to release insulin. Can any of you guess what might tell a cell to release insulin? So this insulin would eventually make its way into the bloodstream. Think about diabetes, things like that. So the signal is glucose. So if there is glucose present, the pancreatic beta cells will read that signal and it will tell them that we need insulin levels to rise. And so the insulin will now be able to leave the cell. And one kind of neat thing about this is that the insulin, I said it's aggregated, it's specially packed in this example. We talked about how there's different types of modifications that can be made at the ER and at the Golgi apparatus. And so one example of a modification made at the Golgi apparatus could be something like this that helps those proteins or molecules to be secreted to pack really tightly so that you kind of get more bang for your buck in an individual vesicle. And again, in regulated secretion, this is a good method if you need to have a really controlled and rapid response to some type of signal. Let's talk about endocytosis. Right? So we've talked about exocytosis, things exiting the cell. Let's talk now about endocytosis, things entering the cell. So one of the most kind of fun examples of endocytosis is a type of endocytosis called phagocytosis, right? And so this is when a cell will engulf a very large particle. On the left figure, we have an example of a white blood cell. So white blood cells, they're immune cells. They go around and they hunt down bacteria. And here we have an example of a white blood cell phagocytizing a bacterium, right? So what it does is it shoves out extensions of its plasma membrane called pseudopods to surround that bacteria. Now that bacteria has a little membrane around it, and then it's going to be it's going to be digested by the cell. On the right side in B, we have an example, kind of a different perspective, looking at a cell that is phagocytosing another cell, and you can see it's got kind of this it's a very thin layer of the cell that it's wrapping around the target, which is kind of creepy looking. Now, let's think back about kind of the different parts of the cell. What is it that allows this cell this white blood cell to extend pseudopods, right? Think about that for a minute. What parts of the cell would control that? So hopefully you've been thinking about cytoskeleton, right? So the actin cytoskeleton is right under the surface of the cell membrane, and that is going to rearrange to help to push out these pseudopods, right? To help to create these extensions to the cell. Again, the cytoskeleton, it's like the skeleton for the cell. So once we have, for example, this bacterium, it's been engulfed. Now it has a phospholipid bilayer around it, and so it's almost like its own little organelle, and it's called a phagosome at this point. And so this phagosome needs to digest that bacteria, and so what will happen is it will fuse with a pre-existing lysosome in the cell, and then the lysosome components will degrade and digest that bacteria. In addition to our immune response, another example can be seen in protozoans, so these small single-celled organisms, the way that they feed. Now, a little bit more about kind of how this works. There has to be some type of signal for that cell to extend these pseudopods. Something has to tell them that this is food, right? And so what the cell has is receptors on the surface, and if those receptors bind to the correct targets, it will induce the creation of these pseudopods and engulfing of that particle. And so these receptors are very particular, right? The cell shouldn't eat anything and everything that it comes into contact with. So one example of a signal or a marker that will bind to the surface receptors and tell the cell, such as the immune cell, that this is something it should eat would be an antibody, right? Our immune system produces antibodies in response to foreign bodies or bacteria, and these antibodies stick all over the surface of that particle. And when the receptors on the surface of the cell come into contact with those antibodies, that is the right combination, and that is a signal to the cell to say, yes, this is edible, let's create a pseudopod, let's engulf this thing. It's pretty cool. So another way, or another type of endocytosis is called pinocytosis, right? So this is very nonspecific in what it is grabbing up. We have a picture here on the right. You can see that these inbuddings or in pockets are forming, and whatever happens to be around in the extracellular fluid is what gets taken up into that vesicle. Now this is a constant or constitutive process. It's always happening, right? And again, it's not specific for anything. It is just taking whatever is in the surroundings. Okay, so this is also called cell drinking. because it's just always happening and it's taking up a lot of fluid, right? And you've seen this formation of vesicles before, and sure enough, the mechanisms behind the formation of these vesicles for penocytosis involve clathrin. We've also talked about how a cell can grow its cell membrane, right? We talked about how phospholipids can be synthesized and introduced into the cell membrane of the ER, and a portion of the ER can bud off, travel to the surface of the cell, fuse with the cell membrane, and provide more material. Now, if you need to maybe shrink the cell membrane, make the cell a little bit smaller in one area or overall, the way that the cell does this is penocytosis. So this is a great way to regulate how big the cell membrane is, and also probably to recycle kind of what is at the surface of the cell. Now, we talked about penocytosis as being not very non-specific, right? Now, the most specific method of endocytosis, or another specific method in addition to phagocytosis, is receptor-mediated endocytosis, right? And this should look kind of familiar at the beginning. So just like when we talked about secretion of vesicles, how these vesicles form and the clathrin helps to determine what cargo is in that vesicle, the same way when endocytosis happens, when something from the surrounding needs to be taken into the cell, that something is selected by the same methods. So you can see in this example, we have in the extracellular space, we have cholesterol that is wrapped around LDL, and that package needs to be taken up into the cell via a vesicle. And so we have clathrin that is going to interact with a receptor, and that clathrin is going to help to force this budding spherical shape, as well as going to help specify what is going to be in this vesicle that's being formed. And those receptors there's a variety of different receptors, and each receptor type is specific to a different cargo. And so in this example, the clathrin has recruited LDL receptors, and the LDL receptors will only bind to LDL. So if there's any LDL outside of the cell, it's going to bind to that receptor, get taken into that forming vesicle, and endocytosed into the cell. Right? Do you remember what helps that vesicle to bud off? Dynamin, right? So the clathrin-coated vesicle, once it's formed and released from the cell membrane, is going to lose that clathrin coat. And now we've got this cholesterol, this LDL inside of the cell, and the cell wants to use it for something, right? So what will happen is that vesicle will travel to an endosome. It will fuse with the endosome and release its contents into that organelle. Whatever is in the lumen is going to be released into the lumen of the endosome, and whatever is in the whatever is in that plasma membrane of that vesicle is now going to be incorporated into the plasma membrane of that endosome, as you can see in that blue receptor. So in order to further break down that LDL and cholesterol, the endosome will it will travel to a lysosome, and it'll fuse with that lysosome. And the lysosome is where lots of things can get broken down, right? And so here we can see enzymes are breaking down that LDL in releasing the cholesterol from that little package that it is in. And now that cholesterol can be used by the cell. And the cell's pretty smart, it recycles, so we can see that the receptor for LDL, it does not get destroyed, but in fact, a vesicle containing the receptor in its membrane is going to bud off of the endosome, and it's going to travel back out to the cell surface, fuse with the surface of the cell, and now we have that LDL receptor ready to go again. So, receptor-mediated endocytosis, if it is endocytosis being mediated by or occurring because of receptors. And anytime you hear about receptors, you always think those are specific, right? And so receptor-mediated endocytosis is implying that you're taking up very specific things that are desired by the cell from its surroundings. I encourage you to watch a video that demonstrates this in more detail and a little more exciting by following this link. So I'm going to skip over the part about the acidic endosome. It's just acidic to help, kind of start breaking things down. But I want to talk about, what do you think this cholesterol could be used for? Right? So why would a cell want to take up cholesterol? We've talked about cholesterol in previous chapters. So cholesterol can be inserted into the cell membrane, and it can help regulate fluidity of the cell membrane, right? To make it stiffer or more flexible or more fluid, right? Now let's talk a little bit more about recycling, right? So endosomes are great for recycling things in the cell. So here we have an example of a cell that has, on the apical side, it has endocytose something. So we've got a little vesicle that contains some particles, and it's going to travel first to the early endosome. Right? So depending on what is in that vesicle, we can have a variety of things happen. So for example, that vesicle or those particles that are in the early endosome could bud off, and that vesicle could travel to a lysosome and the contents could be broken down. So that would be that point 2. We've also talked about recycling, right? So say you had some type of receptor that was involved in endocytosis. It'll travel to the early endosome and then be sent back out via a vesicle to the cell surface again. An alternative method in 3 is called transcytosis. So maybe you have a receptor that was on the surface of the cell on the apical plasma membrane, and you want to move it to, say, the lateral, the basolateral, so basal on the bottom or lateral on the side, right? So if you want to rearrange things on the surface of the cell. In this example, you can see that there are some tight junctions, right? Those tight junctions are going to prevent movement of things in the plasma membrane. So right now, if there was a receptor or a transporter on the apical side of the cell, on the apical surface, it could migrate back and forth, but once it gets to that tight junction, it's going to bump back and go in the other direction, right? There's no way for that transporter or that transmembrane protein to get to other sides of the cell. So if the cell needs to move that protein that's inserted in the apical membrane, what it will do is transcytosis. It will endocytose that receptor, that transmembrane protein. That vesicle will go to the endosome, and then that vesicle, instead of going back up to the apical membrane, can go to either the lateral or the basal membranes, which is pretty neat. Okay. Again, the endosome has an acidic interior that helps to kind of break down components that have been endocytosed. And also, in addition to components kind of carried inside of the vesicle, we've talked in this slide about receptors and how they can be recycled, they could be degraded, or they could be moved around the cell in a method called transcytosis. The cargo typically goes straight to the lysosome to be degraded. Lysosomes. We've talked about lysosomes before. They are responsible for digesting proteins, and so they have a very low pH, right? So why would a high acidity be important? A high acidity helps to break down proteins. In addition to that, and you can see right inside this lysosome, the pH is about 5, compared to in the cytosol of the cell, which is neutral at about 7. We also have hydrolases, these enzymes that are going to help to break apart proteins and other molecules. So lipases can break apart fats, glycosidases can break down sugars, sulfatases, nucleases, right? We've got all sorts of different aces inside of this lysosome. One thing that's really neat is these acid hydrolases only function at a very high pH. So that is really good as a really good protective mechanism for the cell. If, for example, somehow these enzymes got out of the lysosome, they're not going to wreak havoc on the cell and digesting everything in sight. So when we've talked about enzymes, their catalytic efficiency, how well they can catalyze reactions, can be regulated by things like temperature, by things such as phosphorylation, right, whether it's on or off, and pH is as well another regulatory method. In order to maintain that pH, you see here we have a hydrogen pump. Right? It's using the power of ATP to push hydrogen into this lysosome against its concentration gradient. So this is called what we talked about as active transport. Right? So that ATP-driven hydrogen pump helps to maintain that acidic environment. We also have a metabolite transporter. Think about what that could do. Metabolite and transporter. We see that these metabolites are being transported out of the lysosome. So a metabolite would be basically a particle, a piece of something, of a molecule that you've pieces of a molecule that you've broken apart. Right? So we've talked in the past about, what happens if you degrade this amino acid. What can it be used for? It can be, say, for example, put into the citric acid cycle, right? So that is a metabolite, partially broken down molecule. So as the lysosome is breaking molecules down, whether it's proteins or fats or DNA, it now has all these pieces, these building blocks, also known as metabolites, and they are going to be sent out of the lysosome by this transporter so that they can be used to have different functions and be used to build things in the cell. Now in this, this slide, I really want to emphasize that there are 3 methods for degradation in lysosomes, right? So if something's going to be degraded by a lysosome, there are 3 ways that it gets to that final destination, the lysosome. The first we talked about is phagocytosis, right? So in this example, we have a little bacteria that's been phagocytosed. It now is enclosed in a phagosome. That phagosome is going to then basically fuse with a lysosome, and the lysosome will break down that bacteria. The second way things can get to a lysosome is by endocytosis. So a vesicle forms on the surface of the cell. In this example, maybe it is receptor-mediated endocytosis, maybe it's penocytosis, it could be either. And the contents that are in those vesicles are going to travel to the early endosome. That might mature into the late endosome, and then it will fuse with the lysosome. And again, the lysosome can break down those endocytosed particles. The third method is autophagy. Autophagy, auto, self, right? So in this case, maybe you have something in the cell that you don't need anymore. For example, maybe a mitochondria is old and new or not working and the cell needs to get rid of it. What the cell will do is it will form an autophagosome. So a phospholipid bilayer will form around that organelle to be destroyed. Then that autophagosome will fuse with the lysosome, and the contents, such as in this case a mitochondria, will be broken down. Right? So autophagy and phagocytosis are quite similar. Again, you should be able to compare and contrast these 3 methods, these 3 paths that materials can take on their way to the lysosome for degradation. So that's it for this chapter. The second lecture is pretty short because I wanted to make sure to give you time. Normally we would be working on that worksheet in class. I will take a look on Thursday at some of the questions you want to go over on the worksheet, and I'll probably post a video or a page going over a few of those for you guys. I'm sorry again that it took so long to get your exam scores out, but I have those done. If you would like to look at your exam, just let me know and I'll make sure that you can look at it from home. Also, what did I want to mention? Exams. We will have another exam coming up since we finished chapter 15, but I wanted to make sure to get back to you your second exam, and also some of the homework assignments that you've been doing. So I'm working on getting those homework assignments back to you as fast as I can, and then we will put out a survey in terms of different exam dates for exam 3 coming up. All right, if you have any questions about lab, about Lit Lab, please feel free to email me as usual, and I will see you around. Bye.